about to peer into your future. Why? Because I wrote the test, right? So there are 25 multiple choice questions, which vary in their difficulty. Please do not overthink ones that seem easy. There are some that are just straight up easy, right? Then there's going to be a labeling section, right? We went over lots of different parts of the brain, right? We did. And so, and the things that I spent a great deal of time discussing, I think it's legitimate for me to ask you, what is this structure? What does it do? So we'll go, and we went over neuroanatomy a lot. So we're going to cover neuroanatomy a lot in today's review session. So there's a labeling section where you not only have to label the parts of the brain, but also what they do. Right? So let's say if I showed you a picture of something that looked sort of like a coiled up horn, and I told you it was a part of the forebrain, and it was a subcortical structure, looks sort of like a horn on the inside of the brain, those are the basal ganglia. Right? They look like coiled up horns on the inside. And then you say, basal ganglia, important for the control of motor movement. Be an example of how you would label that part of the brain as well as tell me what it does. Then there is a matching section where you will be matching something to something else. Then there's a fill in the blank portion of the test where there's like a paragraph, and if you really understand the concept that the fill in the blank paragraph is describing, it should be fairly easy for you to pick out the right word to put in there. Right?
Do you give us no.
This is fairly straightforward. You either know about this process or you don't. Then there are 2 essay questions worth 5 points each.
And we have to answer both.
You do. But they're not super long essays. They're not like you'll need another test booklet. You write directly on the exam itself, and I give you a reasonable amount of space with which to this. And it's not like compare and contrast or reflect on. It's please describe this. And then you describe it. And then I tell you exactly what I'm looking for in that description.
Okay.
Please read each question carefully. That's the one thing that students miss points on the most, is they don't read the question. For example, if I ask you for location, function, and structure, please tell me where it is. Please tell me what it's made of. And also tell me what it does. Right? And I will usually bold those words for you, because I'm looking for all of that information. Does that make sense? Read each question carefully and give me everything that I'm asking for to the extent that you can. All right, so who can talk to me about, and now I'm going to sit down because I really don't need a lectern.
Who can talk to me what? You were pushing it there.
Yeah, so talk to me about what is generalization and reductionism? How are they related? Who can remember what they are? They're basics of science. Basics of science, not just this kind of science, but any kind. Yeah?
Reductionism?
Reductionism.
Yeah, reductionism is where they focus on one thing rather than the whole.
Okay, so give me an example of somebody, like you meet a scientist in an elevator and you go, what do you do? Give me an example, somebody, not you necessarily, because that was totally right. Give me an example of somebody describing to you what they do, and they're really focusing on the reduction piece of their science. Yeah?
So they would be specifying on a certain.
Great, give me an example of that.
Let's say I am studying cancer.
Okay.
But I want to know about one particular type of cancer, and then I want to know what is causing that particular type of cancer as opposed to this type of cancer.
By focusing on maybe this particular DNA or
Right,
whatever.
right.
Yeah, that's reductionism because it's something specific. She's describing the specific thing she studies. So be able to recognize examples of somebody describing to you the reduction of their science. I study but look for words like specific, right? I study the this, and it's giving a very highly detailed, specific answer about the nature of their research. So what about generalization? How would you recognize generalization? Yeah?
Cancer as a whole, let's say. Cancer, the reason I know this is because I had cancer, so there's various different kinds of treatments for various different kinds of cancer. But if you wanted to know about the perhaps the beginning of treatment in a cancer.
So let me stop you. So generalization in science is taking something you find in situation A and applying it in situation B. So it's not necessarily going, let's look at cancer as a whole. That's not what we mean by generalization. That's intuitive for you to go, well, if specialization is focused on something, or excuse me, if reductionism is let's focus on something specific, then you want to say, well, it's the opposite of that, generalization, but it's not. Generalization is going, I'm going to study cancer in mice and generalize it to people.
Okay.
I'm going to study treatments for cancer in leukemia and then try to generalize that to brain cancer. Does that make sense? Generalization is taking whatever you find in the lab and generalizing it to a completely different situation. That is generalization. Is that clear?
Yeah.
Right? And the most common way neuropsychologists do this is using what kind of models? What do we usually generalize to people? Animals, right? Your book makes several pages to apologize for animal research. And so that is one of the most common ways that neuropsychologists study human behavior is by using animal models. And they're engaged in generalization when they do that. And they generalize what they see in animals to people. Is that distinction clear? Good. So be able to recognize reductionism and generalization if you see it. So who can tell me what specialization of function is? What is specialization of function? What does that mean?
Well, it's like different areas of the brain specialize in a specific thing.
A specific task.
A specific task.
Or function, absolutely. So that's the idea. And so, and we have some examples of this, right? There are many examples in class. We learned about all the different structures of the brain in neuroanatomy and how each one was responsible for primarily responsible for a specific type of task. So who can give me an example of a part of the brain and what specialization of function it has?
The optic.
The limbic system?
The limbic system is a system that specializes in what? In emotional processing and emotional behavior, really. And there's three structures that make up the limbic system, right? There's portions of the cerebral cortex. It's called the limbic cortex. There's the amygdala and the hippocampus, right? And there's even further specialization of function, right? Because what does the amygdala do? What does it specialize in? Right, emotion. But we talked about in class that there's what emotion has been studied the most in regard to the amygdala? All emotions?
Fear?
Yeah, fear-based responding. So, and the best research suggests that the amygdala seems to be regulating strong emotional responses, particularly negative ones. Although it's also complicit and involved in other emotional responses, where it seems to be the most active and the most involved is when you have strong negative reactions, strong negative emotions. The amygdala seems to be very involved in the regulation of those behaviors. Strong negative responses. Right? Good, so be able to recognize specialization of function. How did they come to know that, well, like we talked about in the very first lecture we had, we talked about how they first started to discover that the brain has specialization of function. How did they figure that out? Well, this part of the brain does this. What do you know? So after the person had passed away, right? But so what had happened to the person? Yeah.
Brain damage?
Yeah, there was a medical doctors way back in the day. They started noticing that people who had suffered stroke or traumatic brain injury had patterns of brain damage in common. Right, the same parts of the brain were damaged when they had the same type of loss of function. One of the first areas that they discovered was Broca's area, right? Broca's area. A series of stroke patients who had donated their bodies to science, right? They saw there was the same area over and over and over again had severe damage when people lost the ability to speak, to utter language. Broca's area. That is an example of specialization of function, right? So again, be able to recognize a description of specialization of function. So then talk to me about neurons, right? right? What are the different parts of the neurons? Because I promise you, you need to know all the different parts of the neurons, what they do, what role they play in a neuron firing or not firing.
Dendrites.
Okay, so they're dendrites. What are the dendrites?
The tree-like branches.
Yeah, tree-like branches. They're connected to, what are they connected to? The soma. And what is their dedicated function? To receive messages. Because what are concentrated on dendrites? What little structures are concentrated on dendrites? Receptor sites, that's right. Now what are receptor sites made out of? So the membrane is made out of lipids. The membrane, the entire skin of a neuron is the membrane. It is made out of lipids. But receptor sites are actually protein structures. Remember that? Protein structures. And then on a receptor site is a molecule called a binding site, right? And so there are receptor sites on the entire membrane, right? There are places where synapses can form on the entire membrane, but they're concentrated on the dendrites. And those little dendrites reach out to try to make synapses with other neurons, right? So we got dendrites. Somebody mentioned that the dendrites are connected to the soma. What in the world is the soma? It's like the body of the neuron, right? It's the big part of the neuron. And what's inside the soma? The nucleus. And what's inside the nucleus? DNA. Right? And so the dendrites are like the ears, and they are dedicated receivers of information. The soma is like the body, contains the DNA, which is all the instructions for the cell. On dendrites are receptor sites that take in messages. Those messages are neurotransmitters, right? All right, so we've got the soma, the membrane. What other parts of the neuron are they?
Ion channels.
The which one?
Ion channels.
So ion channels, yes, another protein structure that lives inside the membrane are ion channels. Super essential for lots and lots of neuronal functioning. Ion channels. What are ion channels? Oh, they come in different varieties, yes. Potassium, sodium, chloride, calcium. There's lots of different types of ion channels. What are they for?
So the ions enter, like the sodium enter, right?
Yes, a sodium ion channel is a particular type of ion channel, and it's a hallway that opens so that sodium ions can get in, right? Through the force of diffusion. Sodium ion channel opens, and sodium's like, woohoo, and rushes into the neuron. Potassium ion channels are just the reverse. They open, and they allow potassium to rush out. There are chloride ion channels, but for the purposes of this class, we're focusing on sodium and potassium, because those are the two big stories, right? So there are ion channels, protein structures that live in the membrane. What else?
Axon.
The axon itself, right? The axon itself, which is where the action potential actually occurs. And what do I mean by that? What is happening to a neuron when an action potential is occurring? It is depolarizing, yes, that is true. What is making it depolarize? Yeah, sodium ion channels opening, right? So in order for a cell to fire, it has to first depolarize. But then once it's firing, it shoots right past zero, which would be complete depolarization, and there's a net positive charge down, right? And that's happening at the axon. It's like a domino effect. Right? There's a particular place on the axon that if it reaches -40, that place is called the axon hillock. When it reaches -40, then all of the sodium ion channels open, one after the other, first at the axon hillock, and then closer and closer and closer and closer to the terminal buttons. In that order, those sodium ion channels are flying open, allowing all the sodium that's outside the neuron to come rushing in. So it stops being -40 and becomes +40 in a particular direction. And when that +40 reaches this last structure that I want to talk about, then that neuron releases its neurotransmitter. Because what's the last structure? We have the terminal buttons, right? Terminal buttons are the end of the axon, and when the sodium ion channels there fly open so that there reaches positive 40, that triggers the release of the neurotransmitters from the terminal buttons, right? I would understand the process of a neuron firing backwards and forwards and upside down. I would, were I you. All right, so where do neurotransmitters get stored when they're waiting to be released? In vesicles, so right, in vesicles. They get stored in vesicles. And when they get released, where do they dump into?
Yeah.
In the synaptic cleft, right? When the neurotransmitters are released, the first place they go is nowhere in particular, into the synaptic cleft. They go into empty space, the little empty space in between two neurons called a synapse, right? And when we're talking about a synapse, we don't really say a terminal button talks to a dendrite. We start talking in terms of membranes, right? So what kind of membrane does a neurotransmitter get released from? The presynaptic membrane, right? Which is the membrane of a terminal button. So the neurotransmitter gets dumped into the synaptic cleft from the presynaptic membrane. It floats across the synapse and then ends up in what kind of membrane? A postsynaptic membrane, which a postsynaptic membrane then has a receptor site, right? And that receptor site has a binding site, right? So all of that you need to know. So, an electrical event triggers a neuron to fire, right? It's this changes in postsynaptic potential, this changes in charge, right? A postsynaptic potential is just a fancy way of saying an ion channel opened, so there was a change in charge. A postsynaptic potential. That's all it means. They're either excitatory or inhibitory. What kind of ion channel opens if it's going to be an excitatory postsynaptic potential? Sodium, right? Because sodium depolarizes the neuron because it's positive. And if positive stuff gets inside, that's going to turn a negative 70 charge into something less negative than negative 70, right? Right. So sodium ion channels depolarize the neuron, but is one sodium ion channel opening going to cause a neuron to fire?
No.
No, but it does make it less negative. It makes it a little more excited, a little more likely to fire, right? All right, so what happens when potassium ion channels open? Yeah, the neuron is becoming hyperpolarized because positive potassium is escaping. So now the neuron is less or more likely to fire? Less likely to fire. This would be inhibitory, right? Sodium ion channels have an excitatory effect on neurons. Potassium ion channels have an inhibitory effect on neurons. And so when at rest, what charge is a neuron at?
Negative 70.
Negative 70. And what does it have to reach in order to fire?
Negative 40.
Negative 40. What do we call negative 40? The threshold of what? Excitation. If a neuron reaches negative 40 at the axon hillock, what happens? It fires. If it doesn't reach negative 40, will it fire? No, it either fires or it doesn't. Are there bigger versus smaller action potentials? Or are action potentials the same every time?
No, they're different.
It's the same.
They're the same each and every time. Every time a neuron fires, it fires the exact same way. It either fires or it doesn't. It's like a light switch. It's not a dimmer switch. It's on or off. And so bigger steps, this is called the all or none principle, right? And our neuron either fires or it doesn't, and every time it fires in the exact same way. And so bigger stimuli make neurons fire not bigger, but more often, right? If a neuron keeps getting excitatory signals, it doesn't mean it has bigger action potentials, it just means it has more of them. It's called the rate law, right? Okay, so an electrical event does trigger the release of the the release of the neurotransmitter, right? Positive 40 reaches the terminal buttons, and that triggers the release of that neurotransmitter. But does electricity, like, leap across the synapse? Well, does it? Did we ever describe the ions flying across the synapse?
No.
No. What flies across the synapse? What actually is the communication from one neuron to another? A neurotransmitter. Now, is a neurotransmitter electrical?
No.
No. It's a chemical, right? So the actual communication between neurons, is it chemical or electrical? It's chemical. Neurons are both electrical and chemical, but the communication itself is a chemical communication. Right? Because it's the neurotransmitters that open up ion channels. That's what they do. That is what neurotransmitters do. That's all they do. All neurotransmitters do is open ion channels. That is their sole function. And when they open ion channels, that either makes a neuron more likely to fire, sodium ion channels, or less likely to fire, potassium ion channels. Now, there are other types of ion channels, just so if you go on and take advanced neuropsychology, right, they're not like, wait a minute, she only told me about sodium and potassium. There actually are other types of ion channels. But we're going to focus on these big two, because these are the two most important, right? Sodium makes a neuron more likely to fire. Potassium ion channels make a neuron less likely to fire. Yeah? Good. So, what are what are neurons then evolved to do? What is a neuron's sole function? To communicate with other neurons, right? The neurons are how your body and your brain communicate with each other and controls itself. All of that communication is happening through neurons. That's it. Neurons are specialized for communication. Sending information around your All right, so there's one other structure in a neuron that the way I sort of understand it is it's not as important for an action potential, although without it, you're never going to get back to the resting state. So they're sort of important to get back to the resting state so a neuron can fire again. There's one other structure we have not discussed yet today. What's another structure inside of a neuron? Also lives in the membrane.
Pumps.
The pumps. Do not confuse the pumps with the channels. I know they both have the word ion in them, and it can become very seductive to think of ion channels and sodium, potassium ion pumps as the same thing, but they're completely different. They have similarities. They both live in the membrane, right? They both are made out of proteins. But they have and they both deal with ions. So I can see how some students might get these two structures confused. But they're different, right? Because ion channels specialize. There are sodium ion channels, which are different than potassium ion channels, right? Different hallways. But sodium potassium ion transporters, or pumps, are different. What do?
They push.
One person, raise your hand.
They bring in 3 sodiums and push out 2 potassium, or the other way around.
So where is sodium and potassium when a neuron is at rest? Start there. Where is sodium concentrated when a neuron is just chilling?
Inside.
Sodium?
Oh,
Outside. Remember, salt water. Sodium chloride outside. If you can remember that sodium chloride is salt.
Okay.
Right? Sodium is outside the neuron. And that's where it ought to be if a neuron is at rest, right? That sodium needs to be outside. Where is potassium when a neuron is at rest? Inside. If you can remember that, then these pumps make a whole lot more sense. Because the pumps are important for maintaining that resting potential. Keeping that sodium outside and keeping that potassium inside. So do these pumps push sodium out? Yes or no? Yes. Do they pull potassium in?
Yes.
Yes, that's what they do. They push sodium out and they pull it in because they're trying to maintain the resting state.
Mm-hmm.
But sodium they're tiny ions. Ions are just atoms, right? They just slip across that membrane. They're like, ha, you're just a lipid membrane. I'm just going to slip across. And those pumps are responsible for pushing that sodium out and pulling that potassium in. But as you point out, there's a ratio. They don't do this in equal amounts, right? So what is the ratio?
They take out 2 and bring in, take out 3 and bring in 2.
Right. So they push 3 sodiums out for every 2 potassiums they bring back in. Does that make sense? So they're constantly pushing more positive out than they're bringing negative in. I would understand the sodium-potassium ion pumps super well. I would know that ratio. I would know where they are. I would know what they're made of and how they're responsible, highly involved in maintaining a resting potential. I would focus a considerable amount of my time on these structures if I were you. But they're not the same thing as ion channels, right? And so when I was first learning about this, I sort of thought to myself, where ion channels seem to be super important for whether or not a neuron is going to have an action potential or not, right? So really deals with action potentials, ion channels. Where sodium-potassium ion transporters or pumps really are more important for the resting potential. Does that make sense? Now they're both needed for all of it, right? But in terms of which is, where I would sort of file this structure, well, I would file ion channels under action potential, yes or no? And I would file pumps under, let's maintain that resting potential. What charge is that resting potential again?
-7.
And there's a word that we use to describe that. Well, 2 words actually.
Resting potential.
Dynamic equilibrium, right? It's another thing we use to describe this. That when these sodium-potassium ion transporters are constantly pumping out sodium and pumping in potassium, that -70 is being maintained, and we call that a dynamic equilibrium. Dynamic equilibrium.
Is the resting potential.
Well, the resting potential is -70, because potential is a fancy word for a charge, right? But the process here, this is being maintained, right? We call the fact that it's being maintained by these active pumps a state of dynamic equilibrium. The charge itself is -70. And remember, potential is a fancy word for charge. That should help you remember which is which. It's in a state of dynamic equilibrium, and the charge, the resting potential, is at -70. Does that make sense? Understand those structures. Yes?
So what happens at -40 and then what happens at +40?
Well, -40 triggers something to happen. So I believe we went over this, like we drew it on the board, right?
I thought you just said the fire is at negative.
Well, it does, right? A neuron will fire when the axon hillock reaches -40. But firing isn't like some magical, mystical event. Firing is, so you can think of sodium ion channels really have 2 roles. One is to get it to -40 at all, right? So ion channel over here opening, ion channel over here opening, it gets to -50, it gets to -49, all these sodium ion channels are opening one at a time, right? Because neurotransmitters at lots of synapses are open sodium ion channels, right? If enough sodium ion channels open so that the neuron becomes -40 at the axon hillock, the axon hillock then goes, okay, all you sodium ion channels on the axon, open. And it kicks open the first door. Sodium ion channel opens and lets lots of sodium in. Then the next door opens. More sodium ions. Next door opens. More sodium ions. And now it's not one at a time, right? It's like bam, 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 bam. And because all of these sodium ion channels are opening, it doesn't stay at -40. Now it shoots up to +40. And this is the action potential. The fact that it's getting to +40 is now a positive electrical event that is occurring. That positive electrical event of +40 is the action potential. Does that make sense? And when that reaches the terminal buttons, it triggers the release of the neurotransmitter. And then the neuron goes back to its resting state, -70 again. And then it may fire again, or not. Depends if it's getting more excitatory impulses or not. Right? Right. Does that make sense? Did I answer your question? All right. And these changes in charge are really short, right? When a neurotransmitter opens up an ion channel, that by itself is nothing, right? There's going to have to be lots of ion channels opening if a neuron is going to depolarize enough to fire. What keeps these changes in charge, these changes in potential, these postsynaptic potentials, really short? What keeps them short? There's two ways and it's essentially, another way of thinking about it, they're kept short by making sure there's not too much neurotransmitter in the synapse. Yeah, reuptake and? Enzymatic deactivation, right? Because that's how your brain keeps these changes in charge short. So that it has to be a lot of input into a neuron to cause it to fire, right? So it keeps the amount of neurotransmitter, your brain, your neurons, ensure that the amount of neurotransmitter in a synapse is small. And the two ways that your brain ensures that, two ways you evolve to ensure that, is one is reuptake. Terminal buttons literally suck back up some of the neurotransmitters that they just released. That process is called reuptake. And is monitored by another type of receptor called an autoreceptor. The other way is through enzymatic deactivation. If any of that neurotransmitter should happen to escape the synapse, there are chemicals waiting there to rip apart those neurotransmitters, called enzymes. So neurotransmitters, if they should leave the synapse, are enzymatically deactivated. Right? So know that. All right. So definitely spend time with the neuron, right? All the parts of the neuron, what a neuron does, how an action potential occurs, what the resting potential occurs. But know all the different types of terminology we use for this, right? Don't just memorize terminal button, you should also know what a presynaptic membrane is, right? All of those different terms are going to be really important. All right, so but we also did a lot of neuroanatomy, right? Large neuroanatomy. And we talked about lots of different brain structures. Now, what's the most basic gross, so gross neuroanatomy doesn't mean it's disgusting, right? It just means big structures. Right, so we focused on the little baby tiny structures, receptor sites, dendrites, membranes, ions, right? But we also, then we sort of shifted gears. We talked about big structures in the brain and big structures in your whole nervous system, right? So if you're going to think about your nervous system as a whole, we can split it up into two separate nervous systems. What are they?
The central nervous system and the PNS.
Yeah, the peripheral nervous system and the central nervous system. So what makes up the central nervous system? The brain and the spinal cord, right? That's the central nervous system. The peripheral nervous system can also be split up into different parts, and it can be split up into the somatic portion of the peripheral nervous system and then the autonomic portion of the peripheral nervous system, right? So understand those sort of levels of specificity, right? The first basic distinction we can make is between central and peripheral. Then we can split up each of those into its separate structures, right? So spend some time being able to do that. So we focused first on the central nervous system, right? And we can split that up into the brain and the spinal cord, right? Okay, so let's just focus on the brain. There's three big parts of the brain. What are they?
Frontal.
The forebrain, the midbrain, and the hindbrain. That's sort of so we've split out the central nervous system into the brain and the spinal cord, right? Now we're focusing on just the brain. We can split the brain up now into three parts: forebrain, midbrain, and hindbrain, right? And we spend the most amount of time talking about the forebrain. Why? Because it's my favorite part.
It's fun.
Well, because it's the mammalian brain, right? The forebrain is the mammalian brain. Take out the brains of mammals, all mammals have a forebrain that have a lot of similarity. A lot of similarity. The midbrain, all right, but then there's the hindbrain, and I call that the lizard brain, right? The lizard brain. Because if I was going to do dissection on a human brain, and I carefully took out the hindbrain and I put it on the table, and then I did the same thing with, I don't know, like a Komodo dragon, reptile, right? And I took out that Komodo dragon's hindbrain, would there be a lot of similarities? What do you think? If human beings have a lizard brain,
Oh, yeah.
do you think that my hindbrain looks very similar to a lizard's hindbrain? And in fact, they do. The hindbrains of various animal species look very similar because we evolved. And the hindbrain, when it evolved, it's not as if the brain was like, well, we're going to become, it's not as through the process of evolution, the hindbrain stopped needing to be structured that way. The same process as in a lizard for respiration, heartbeat, is the same. Lungs, heart. And so the hindbrain is very similar from species to species. And so in terms of evolution, which one is the oldest? The hindbrain. Right? Look at the fossil record. We see brains starting to develop. The hindbrain is the type of structure that seems to have evolved first. Right? It's the evolutionarily oldest, if that makes sense. Very similar, right? The hindbrain. And so but then with the forebrain is where it has many structures itself, right? So what's the most basic way we can split up the forebrain? Into 4 sections. The lobes, right? What are the 4 lobes? Well, we can split the forebrain up into cerebral cortex and then subcortical structures, right? But the cerebral cortex can be split up into 4 lobes, right? What are they? Frontal lobe, occipital lobe, temporal lobe, parietal lobe. All right. So where are they? Where are those 4 lobes, and what in the world do they do? So let's say if I was going to draw a picture of a brain, what would I put in the front? Frontal lobe. What would I put on the top?
The dorsal. Or the
Parietal lobe. Do not confuse the direction words with the lobe words. Right? Yes, the parietal lobe is located in the dorsal section. That's because dorsal means what?
top.
Means top, right? So if I was going to draw a picture of a brain, let's say I was going to draw it from the side, the frontal lobe would be in the front, right?